Welcome back to my course Industrial Biotechnology. Now, in the last lecture, I was discussing about the penicillin fermentation process. The use of penicillin actually started after the Second World War because penicillin is a narrow spectrum antibiotics, and it is active against the gram-positive bacteria. And penicillin actually it inhibits the cell wall formation, and that is how the cell killed. But it doesn't doesn't affect our intercellular component present in our system. So now penicillin is the first organism, that microorganism that was discovered which produces penicillin, that is Penicillium notatum, and this Penicillium notatum, after that we switch over to the Penicillium chrysogenum for the improvement of the penicillin production. Now, for penicillin production, it is similar as compared to the citric acid fermentation process. Here also you have to prepare the culture in the form of in the lab you have to prepare the culture in the form of spores, and then spores you put it in the seed tank. After seed tank, you bring it to the fermentation plant, and then you transfer to the inoculum vessel where the vegetative cell will form, and this vegetative cell you transfer to the production fermenter. And then the production fermenter, after the usually the fermentation time in the inoculum vessel, it varies from 30 to maybe 40 hours. And in case of production fermenter, it is about 125 to 140 hours or 44 hours. So, it required different carbon source, different nitrogen source. Corn-step liquor was found very good nitrogen source as per penicillin production is concerned, and as per carbon source is concerned, lactose is found very important carbon source, and glucose is used as a source of energy. And but I told you that as it has little difference as compared to other fermentation process. The reason is that it required the precursor. As for example, for penicillin G production, we required phenyl acetic acid. In case of penicillin V formation, we required phenoxyacetic acid. Now, I told you that downstream processing of the penicillin also quite interesting. We shall have to use the solvent extraction process, solvent extraction process to purify the penicillin. Ultimately, we add some kind of potassium salt in it to precipitate out penicillin, potassium or sodium salt in it to precipitate out penicillin in the form of sodium or potassium salt. Now, penicillin is usually marketed in two different forms, either in the form of capsule or in the form of injection fluid. Let me go ahead with this and let me see that how exactly the penicillin produced by the industry, how it is produced. Now, first as I told you that penicillin, it produces beautiful spores. Can you see it? This is a very green spores and very beautiful looking actually. I told you in case of, I have shown you the spores of this Aspergillus niger, that is totally black, but there is a green spores. Now, if you see under microscope, that you will see like this. This is a candiospores. So this is the kind of branches we have. You can see that how Penicillium chrysogenum looks. Now, when penicillin first made at the end of Second World War using the Penicillium notatum, the process made 1 milligram per decimeter cube. 1 decimeter cube is equivalent to 1 liter, because that I have to write it here. dm decimeter cubed is equal to 1 liter. So 1 milligram per liter was the initial concentration of penicillin that was first we found out in the fermentation broth. Now, today you will be surprised to know that by using different species with some genetic modification, better extraction procedure it is possible to increase the penicillin concentration as high as 50 grams per liter or decimeter cubed. This is a constant, there is a constant surge to improve the yield, generating mutants using the UV radiation. Because not only UV radiation, we use different kind of genetic modification we also carry out in the system. The methods increase the penicillin yield, the improvement of the composition of media. By improved composition media, it is possible to increase the penicillin yield. Then isolation of better penicillin, producing molds like Penicillium chrysogenum, which grow better in huge deep fermentation tank, and development of submerged culture technique for the cultivation of mold in large volume liquid media through which the sterile air is forced. So initially because this fungi can grow both in the surface culture and submerged culture. We find for the penicillin fermentation process, submerged culture is best for the penicillin production. Now, industrial production of antibiotic penicillin, industrial production of penicillin broadly classified into two processes. One is called upstream processing. Upstream processes encompasses any technology that leads to the synthesis of product. That the upstream includes the exploration and development and production, and downstream processing, basically the extraction, purification of biotechnological product from the fermentation is referred as the downstream processing. So in a broader sense, I can say that upstream processing, we develop the technology through which we can produce in the penicillin in the fermentation broth, and downstream processing, how this penicillin we can purify it from the fermentation. But I told you that whenever we market any kind of product, it is necessary to purify the product. If you don't purify, then the value, the cost of the product will drastically reduce. If you have more purification, the cost will be more high and it will be more useful. Then upstream processing, the media is designed to provide the organism with the nutrient that is required. So this is very important. I told you not only the microorganism that is very important because microorganism that is used for the industrial fermentation process should be the industrial strain. Industrial strain, it is not only should have the higher productivity. I mentioned that if the product concentration is high, your recovery cost or purification cost will be low, so your cost of the product will reduce drastically. So this is the one that is, and not another is the genetic stability, that biochemical activity of the organism should be same, so that different generation you will get the same product. The productivity in the, or if you use the different generation of organism, that should not change with respect to generation. So this should be very this is very important factor that we have in case of penicillin or any fermentation process. Now, media composition plays very important role for increasing the productivity of the system. So here media composition is to be optimized so that we can get the good amount of this product formation. The inoculation method by submerged culture, I told you submerged culture is the best. The reason is that your organism can grow throughout the liquid. Spores are major source of inoculum because in the lab, if you prepare the culture, is not that culture is not sufficient for the grow the culture in the production fermenter. So we shall have to prepare the spores, so quantify the culture, and then we transfer it to the inoculum vessel where we can get the vegetative cell. That vegetative cell can be used as a inoculum for the production fermenter. In the upstream processing, raw materials required may be different. We have carbon source. We can do lactose acts as a very satisfactory carbon compound provided that is used in a concentration of 6% weight by volume. Others such as glucose and sucrose may be used. Glucose and sucrose can be used as an energy source, and lactose is used as a carbon source. Nitrogen source, as I mentioned, constable liquor appears to be the best carbon source as far penicillin production is concerned. The other nitrogen source are ammonium sulfate, ammonium acetate, can be used also as a nitrogen source. Different minerals that also plays important role, elements namely the potassium, phosphorus, magnesium, sulfur, zinc, copper are essential for the penicillin production. Some of these are applied by the corn liquor, because corn liquor, I told you, corn liquor comprises of different type of material, because not only it contains nitrogen source, it contains some minerals, some kind of vitamins also it contains. So it is comprises of a lot of things. The calcium can be added in the form of chalk to encounter the natural acidity of corn liquor. Corn liquor has some kind of acidic pH, so we use the calcium ion just to neutralize that. Now, I told you the precursor is very much required for the penicillin fermentation process, the phenyl acetic acid and phenoxyacetic acid. Phenyl acetic acid is used for the production of penicillin G, and phenoxyacetic acid is used for the production of penicillin V. Now, in the upstream processing, we have the media is inoculated with suspension of conidia, that is that conidia spores actually, that is Penicillium chrysogenum. The media is constantly aerated and agitated, and the mold grows throughout as pellet. Now, here again let me point out that in case of fungal fermentation process, we use the start tank reactor. Now, in the start tank reactor, we use the mechanical agitator, and it has been observed that if we put the agitator on from the 0 hour, it will affect the growth of the organism to a great extent. So, you have to keep the agitator off for some time, initial phase of fermentation, let the mycelia to build up inside the reactor, and then you put this main purpose of the agitator to keep the cell in suspension, but until unless if they grow properly, we cannot put it on. When it grow properly, then and only then we put the agitator on, make it suspension. So there are different ideal time for the production fermenter and for the inoculum vessel, that varies from the fermentation to fermentation process. After about 7 days, the growth is complete and pH rises to 8 and above, and penicillin production ceases. Because now question comes, how penicillin increases. Penicillin, how the pH increases. pH increases due to the reason, mostly due to the deamination reactions, ammonia formation take place from the protein. When undergo the deamination reaction, is produced the ammonia, and then ammonia formation increases the kind of pH of the media. Now, another thing I forgot to mention, that fed batch reactor is appear to be best for this for the penicillin. Fed batch reactor is appear to be best in case of penicillin fermentation process. The reason is that phenyl acetic acid and phenoxyacetic acid is used as a precursor, and you know that and stoichiometry is less. 1 mole of if you write 1 mole of penicillin G, we required 1 mole of phenyl acetic acid. So what I want to mention here, that that means you know that how much penicillin is to be. So if we, on the basis of stoichiometry, if we add all the phenyl acetic acid at the initial phase of fermentation process, pH immediately will drop down, and that affects the fermentation process. That is why you have to, if you use the phenyl acetic acid in a fed batch way, then and only then it will increase the penicillin production. Let me explain that how the fed batch process works. That you know that suppose there is a level, that there is you find out that substrate concentration, because this fed batch reactor is used in case of substrate inhibition. in case of substrate inhibition, it is used. Suppose the substrate concentration that you find that this level is below the inhibition, above this the inhibition take place. So this concentration will keep on decreasing, then again increase, then again decrease, then again increase, like this. So it is like this. So this is a fermentation process. So you take small media that we have agitated here, and then we with the concentration of the substrate will go down, then again we add another same volume of media, then again the concentration, or you can increase little bit, increase the volume like this, and then so that concentration rise up to this level, again concentration will drop down, again you increase the concentration like this, again it is drop down, again you increase the concentration level. So like this you come here, when it is totally filled up, then you when you your fermentation complete, your product formation keep on rising like this. So but then you stop the fermentation and take out the product from the reactor. So this reactor, phase-bridge reactor, was found most suitable for the penicillin fermentation process, because substrate acts as the inhibitor. After that is, and after the end of the fermentation, pH rises to 8. Fed-batch mode, high glucose inhibition of penicillin production take place. That can be taken care by using the fed-batch reactor. The fermentation conditions is 20 to 24 degrees centigrade, pH 6 to 6.5, pressure higher than the atmospheric pressure 1.02 to prevent the contamination. Because here, let me tell you the very interesting thing, that this is a aerobic fermentation process. So when you do the aeration, always there will be your air is going out, and air in. So if we have more air, there will be some positive pressure. If there is a positive pressure, then there is a less possibility to enter the air from different sources, because if even there is a leak in case of mechanical seal, that the air will not come, because if it is a positive pressure, the air contamination problem can be avoided. That is why it is written there higher than the atmospheric pressure, to prevent the contamination from occurring. And spurging air bubbles to provide the sufficient oxygen, because oxygen is sparingly soluble in water, and spurging rate is about 2.5 cubic meter per minute. It depends on the volume of the fermenter impeller to mix the culture evenly. So it is use the impeller. I have already shown you how it looks. The impeller, it is like this. And if you take the top view or bottom view, it is like this. The disc, in the disc, this is center point, and this is the blades are mounted like this. So this is rotated like this. So and this is called impellers. And this is the fermenters, and this is how the impellers that look. This is the different type of fermenter that is used. We have we can have this different air in. I showed you there is the air in, and this is air in and air out that we have. Air in here and air out, gas out there. That is the temperature monitoring system that we have. Now, after the fermentation is over, we will have to purify the product, which is very important. Let me give you some detailed information on the purification process. Now, downstream processing is relatively easy, since the penicillin secreted into the media, and this is an extracellular product. This is not an intercellular product. So there is no need of break open the fungal cell. However, the product need very pure, and since it is being used for therapeutic medicinal drug, so it is dissolved and then precipitated with the potassium salt to separate it from other substances in the media. I told you by solvent extraction process, the penicillin is purified, and then finally we add some kind of sodium or potassium salt to produce the sodium and potassium salt of penicillin. And then removal of cells. First steps is the removal of steps, that recovery is the separation of the whole cell and other insoluble ingredients of the culture broth. That is the filtration we use. We use the rotary vacuum filter. I already mentioned what is called rotary vacuum filter. We have a drum, and inside that it is embedded with a thin muslin cloth, and one knife just touches on the surface of the muslin cloth, and it is immersed in a trough where your fermentation broth is there, and we applied vacuum in the pipeline so that it suck the water and suck the liquid and the cell with stick on the surface of the muslin cloth. And since when it is touching with the muslin cloth surface, the cell automatically will be dislodged from the surface of the muslin cloth, and we can collect in the collector or in the wagon, relay wagon. We can collect for take it out. Now after taking out this, as I told you, the pH is adjusted to 2 to 2.5 with the help of phosphoric and sulfuric acid, and then in the aqueous solution at low pH there is a partition coefficient in favor of certain organic solvent such as the butyl acetate, amyl acetate. So when you reduce the pH, the partition coefficient will be that the solubility of solute will be more in butyl acetate as compared to the aqueous layer. So most of the penicillin that present in the aqueous layer, that goes to the solvent layer, and to dissolve the penicillin present in the filtrate, the organic solvents such as acetyl butyl are used. And then these steps has to be carried out quickly. For penicillin is very unstable at low pH, because we know penicillin, particular penicillin G, it is very unstable at lower pH. And then centrifuge is done to separate the solid waste from the liquid. Penicillin dissolved in the solvent undergoes series of extraction process to obtain the better purity, because again we increase the pH of some aqueous solution. We use some kind of solution so that the kind of buffer solution, so that penicillin can comes to the aqueous layer, again it goes to the solvent layer. Like this we have the purification of the process. Antibiotics then extracted back to the aqueous buffer at pH 7.5. The partition coefficient now being stronger in favor of aqueous phase. The resulting aqueous phase solution again acidified and re-extracted with the organic solvent. So this is kind of repetition of the process that is going on to get the purification of the product. These shifts between water and solvent help the purification of penicillin. The treatment of crude penicillin extract varies according to the objective, but involves the formation of appropriate penicillin salt. And penicillin is present high concentration in the ether solution, is being mixed with a solution of potassium or sodium acetate to obtain the penicillin or sodium salt of penicillin. So that we can penicillin sodium or potassium salt we can produce if we use the sodium salt or penicillin potassium salt, then we get that. This is followed by charcoal treatment, because we have seen that charcoal has some bleaching. This eliminate the pyrogen and by sterilization process, and the drying is necessary to remove the remaining moisture present in the powder penicillin salt, and pure metal salt of penicillin can be safely sterilized by dry heat if desired, and therefore the aqueous solution of penicillin is subjected to crystallization. Penicillin is stored in a containers and kept in the dry environment. So this is, I told you that particularly penicillin, when it is used in the form of capsule, it is not required that much of stringent condition, but when you use in the form of injection vial, it should be 100% sterile. But since it is used for the therapeutic purpose, it should be as pure as possible. The further processing for parenteral use, the antibiotic is packed in a sterile vial as a powder or suspension. For oral use, it is tableted using the film coating. Then searching test for purity, potency are performed on a appreciable number on random sampling of the finished product. And it must satisfy fully the strict government standard before being marketed. So products, the resulting penicillin can be chemically and enzymatically modified to make the varieties of penicillin which slightly differs in property, because as I mentioned that penicillin G is unstable in the acidic pH, but when you produce the penicillin is converted to ampicillin, this is little bit stable in the acidic pH as compared to the penicillin G. This semisynthetic penicillin includes the penicillin V, penicillin O, ampicillin and amoxicillin. Now, precursor, I told you that penicillin G, we have phenoxyacetic acid. Formula is this is phenyl. Sorry, this is phenyl acetic acid C6H5CH2. The acetic acid formula is CH3COOH. The one hydrogen will remove, and it is C6H5CH2COOH. This is phenyl acetic acid. From that we produce the penicillin G. And this is the potassium salt of penicillin, mostly used in the form of injection vial, and sodium salt actually that is used for maybe in the form of capsule, that is where we use the phenoxyacetic acid. This is used in the form of tablet. This is penicillin V, while the phenoxy, the O group is there. This is CH2, but this is O group is there, that is OH group is there, that is why we call it phenoxy. So this is used for the production of penicillin V. Now, the phenoxymethyl penicillin, the production of penicillin V is the phenoxymethyl penicillin addition of different acyl group to the media, and phenoxyacetic group is the precursor instead of phenyl acetic acid, that is we use. Now, there is some saline feature we have in the penicillin fermentation process. I like to mention that nitrogen source is constip liquor, then energy source is glucose, carbon source is lactose, optimum temperature is 25 to 27 degrees centigrade. The fermentation is aerobic, it is the secondary metabolized products, that how the seed is prepared by sporulation of the fungal strain, but this sporulation we don't use for production fermenter. This we produce the inoculated in the inoculum vessel to get the vegetative cell, and that vegetative cell is used for as the inoculum for the production fermenter. Then when the penicillin is produced after 40 hours of fermentation, and how long this production take place, that is 40 hours. What is the precursor of penicillin G? That is phenyl acetic acid. Now, if I ask you what is the precursor of penicillin V? Phenoxyacetic acid. What was the first fungus known to produce penicillin? That is Penicillium notatum. What is the species produce 6 milligram per liter of penicillin? This is Penicillium chrysogenum. What is the how did the scientists improve the yield still further? By genetic modification. What is the batch culture is used? What is why the batch culture used? The problem of aeration, that is why it is used. And major problem of penicillin fermentation is substrate inhibition, and to improve the substrate, to take care of the substrate inhibition, we can go for the fed batch process, where the substrate inhibition can be taken into account. And then what is the major downstream processing that we have? Liquid extraction, butyl acetate or amyl acetate that we use, precipitation of potassium salt, and aseptic conditions. Why cannot the penicillin G taken orally? Because it is unstable in acidic pH. Name the form of penicillin which can be taken orally, that is penicillin B or ampicillin. How does the penicillin kill the bacteria to stop the production of cell wall, and why the gram-negative bacteria not killed by the penicillin? Because it has the different cell wall. So we can understand that that different use of penicillin, how the penicillin act on the human body, how the production of penicillin take place, which has several we have a huge market in the biochemical industries, and we are greatly benefited due to the penicillin that is produced in the market, available in the market, and this has regular use for curing the different infections, particularly in case of athletics. It is quite regular because they might be having some wounds in the when they fall down, and that causes some kind of pauses, and this usually taken care if we use some kind of penicillin. And penicillin can be used, largely used for different type of curing the different type of infections. So with this, I want to point out that if you look at the extraction process, that if you compare the downstream processing as compared to citric acid production also take place by using fungi, and penicillin also produced by using fungi. In case of citric acid, we use the Aspergillus niger. Penicillin we produce by Penicillium chrysogenum. Both are fungi, but if you look at, and both are the secondary metabolites, and but if you look at the downstream processing, there are a lot of difference in the downstream processing of citric acid. We separated through the process of crystallization. Here we separate the penicillin by solvent extraction process. After that we use some kind of sodium or potassium salt of so that of acetic acid, so acetate to get the sodium and potassium salt of penicillin, and finally we can produce in the form of crystals, dry powder or injection fluid. I hope this information will good enough to understand about the penicillin fermentation process. Thank you.